Great. Well, good morning. So it's my pleasure to introduce Zoe Hilbert, and who's a faculty candidate for the Micro and Immunosearch. And I'm really excited to hear about her work, as I'm sure many of you in the audience are too. One of my New Year's resolutions was to increase efficiencies, so I'm going to keep this intro short and just hit no more than 4 highlights. That's my new goal in introducing anyone, probably without the preamble, because I feel like I'm being counterproductive. So Zoe did her undergraduate work at Columbia and got her PhD with Dennis Kim at MIT, working on C. elegans. She went from there to the University of Utah Health Center to work with Nels Eld, doing some really cool work, both looking at evolution of pathogens and of hosts. And I think that combination is really powerful. 2 things from her CV I'd like to highlight. One was that in 2020, Zoe was a Leading Edge Fellow, which is a really powerful community to increase inclusion and belonging in research. And if you meet with her, I'm sure she'd be happy to talk more about that. And is supported by the Helen Hay Whitney Foundation for her PhD work, which she will talk about in this presentation today. So with that, I welcome Zoe.
Okay, great. Can everyone hear me? Yeah, we're good? Awesome. So thank you, Deb, for that really kind introduction, and I'm super excited to be here after virtually visiting a couple of years ago. It's great to put some faces to the Zoom screens, I guess. So I'll just jump right in and say that I am super kind of fascinated by fungi. And in particular, I'm fascinated by fungal evolution and the evolutionary relationships that fungi establish and maintain with the species around them. And one of the things that I find really fascinating about fungi is just the incredible diversity contained within the fungal kingdom. And I think you can start to appreciate this just by looking at all of the different forms that fungi can take. I'm showing you some of these here. So up here you can see these mushrooms, which we see in our gardens and in our forests and on our pizza. And then we also have microscopic budding yeast like Saccharomyces cerevisiae, which many of us know and love for all of the amazing biology and genetics that we've been able to learn from studying this species in the lab. But fungi can also take on these more exotic and sinister forms, such as these 2 examples down here, which I think really bring us into the realm of how fungi can provoke and live in conflict with the species around them. So right here in the middle, you're seeing one of my favorite fungal species. This is a nematophagous fungi. And what they do is they make these trap-like ring structures out of their hyphae. They secrete chemicals which lure these nematodes into the traps, and then the traps clamp down, trapping the nematodes, and they get their insides sucked out, which provides nutrients for fungal growth. On the bottom is another kind of fan favorite. So these are the zombie ant fungi, which come from the Ophiocordyceps genus or other related species. And these fungi infect insects, and they cause these really fascinating and dramatic behavioral changes, which ultimately increase the dissemination of fungal spores out of these fruiting bodies that sprout from the carcasses of infected insects. And so these kind of illustrate some examples of fungi in conflict, but of course another way that fungi live in conflict with the species around them is by their ability to infect many different types of animals. And so it turns out that there's also an incredible amount of diversity among fungal pathogens. So on this tree here, you're seeing about 1,500 different fungal species. The lineages that are capable of infecting humans in particular are indicated in red on this tree. And I've indicated some of our kind of favorite model fungal pathogens by these icons here. Now, there's a couple things I want you to take away from this tree. First, I want you to appreciate that pathogenicity has actually evolved multiple times across fungal evolution. So we're seeing that these red lineages are really dispersed across the entire phylogeny and not clustering in one place. And so I think this raises really interesting questions about how the evolution of pathogenicity actually occurs, which is a process that we don't really fully understand. And I think this also raises the really kind of intriguing question to me about these closely related but non-pathogenic lineages. Are these just kind of poised on the knife's edge of tipping over into pathogenicity? The other thing that I want you to take away from this phylogeny is really just the sheer amount of diversity contained among fungal pathogens. So down here, I've given you a rough estimate of 100 million years of evolutionary time. And so if you do some kind of visual mapping onto this tree, what I hope you can appreciate is that there's really just an incredible amount of evolutionary diversity across these fungal pathogens. And yet when we encounter infections by these species in the clinic, we tend to, well, we're really kind of limited in our ability to treat them. So we only have a few therapeutic options. And so even though the basic biology of these species is likely very different, we tend to treat them in exactly the same way. And so I think this is really reflected in the mortality that's associated with invasive fungal disease. So here I'm showing you mortality estimates for fungal infections compared to some other major infectious diseases, but these numbers are not entirely separate. So fungal infections in particular are a really big problem for immune-compromised patients, and they're currently estimated to account for about 40 to 50% of HIV and AIDS-related mortality annually. So clearly, these fungi really have a significant impact on human morbidity and mortality. And for other pathogens that cause kind of this level of destruction on human health, like HIV and flu and SARS-CoV-2, so mostly these viral diseases, we tend to think a lot about how these viruses, these causative agents of these diseases have evolved. And I think now is really the time to start thinking about these types of questions in fungal species as well. And so studies of how infectious microbes engage with their hosts and shape the evolution of host responses and vice versa really started with the studies of viral pathogens. And we've seen how viruses and hosts engage in these arms race-like conflicts. And these studies of viral pathogens have really taught us how to identify these conflicts and also have shown us some of the mechanisms of how they play out functionally in these host pathogen interactions. As we've extended this sorts of evolutionary thinking to other types of pathogens, so thinking now about bacterial pathogens, what we've been able to show is that these evolutionary relationships and infectious microbes have really shaped broad biological processes within host species. And so work in the LD lab, as well as many other groups, have revealed that these sorts of conflicts with bacterial pathogens have shaped things like iron receptors or even water balance control in the intestine. And so the work that I've been doing as a postdoc and what will continue to motivate my lab's work moving forward is to really now take this evolutionary lens and to shine it onto the study of fungal pathogens. And I think this is exciting, not just because we can add a new microbe to the mix here, but it's really in the nature of these fungal pathogens. So these are eukaryotes, and the hosts that they infect are also eukaryotes. And so there's a lot of really shared basic biology here, which I think means that the evolutionary conflicts and conversations that are playing out across these interfaces are likely to look really different from what we've seen before with other types of infectious microbes. I think beyond this, taking these evolutionary approaches also gives us the power to learn some really kind of fundamental biology about fungal pathogenesis and host responses to fungal infection, which I think is still a really understudied topic. So my research program has really focused on exploring these questions, as Deb mentioned, from both sides of this interface, so both from the host perspective as well as from the fungal perspective. And so I'll give you a glimpse into this work and tell you about what I've been doing on the host side, looking at a sort of unexpected interface of evolution involving host immune receptors that detect microbial carbohydrates. And then I'll take you on an adventure in fungal world, where I'll tell you about how I've been studying how different types of hosts can drive adaptive evolution in one particular fungal pathogen. We'll start first on the host side, and here really the big question that I wanted to answer was, can we find evidence that fungal pathogens have actually shaped the evolution of their hosts? So can we find these sorts of evolutionary conflicts between hosts and fungal pathogens, and where do they occur? And so when we think about these sorts of evolutionary conflicts, we often think about something that looks like this. So we have a pathogen protein that inhibits the function of a host factor. And the really high fitness consequences associated with these sorts of interactions can drive these cycles of adaptation, followed by counter-adaptation, and so on and so forth, with both sides really trying to gain an advantage over the other. Now, it turns out that these sorts of interactions actually leave scars in the genomes of the species involved. And so we, as researchers, can go in and look for particular signatures of this adaptive evolution, which are usually characterized by the accumulation of non-synonymous amino acid changes to synonymous changes. And so I'll refer to this as signatures of recurrent positive selection or rapid evolution. Now, historically, when we think about these sorts of conflicts, we've really been focused on these protein-protein interfaces. But as I started to think about applying this idea to the study of host-fungal interactions, I decided to look at a really different interface and instead to focus on interactions occurring at the fungal cell wall. And the reason that I decided to do this is really because the fungal cell wall is the fungus's first line of defense against hosts, and it's also the first thing that host immune systems see about these invading microbes. The cell wall, as I very simply diagrammed for you over here, is largely composed of carbohydrates, so complex carbohydrate structures, which I'll also call glycans. And I think this was also a really appealing place to look because we know from looking across different fungal pathogens that the cell wall is really a site that these pathogens diversify. And so this happens across evolutionary timescales, but also in response to host cues. So many fungal pathogens will actually shield their cell wall components or use other mechanisms to prevent detection by host immune systems. Combined with this, we also know that hosts contain a whole arsenal of different factors that are involved in the detection of these cell wall components. So here's a list of about 25 different immune receptors that are involved in the detection of microbial carbohydrates, including those found in fungi. And so what I decided to do is to take this list and to look for these signatures of rapid evolution across 3 different mammalian lineages. So I focused on primates, rodents, and bats. And so if we start first with the primates, the way that I've plotted this data is as a heat map. So genes with no evidence of these sort of telltale signs of host pathogen conflicts are indicated in this lightest shade of blue. And then as we progress to the darkest shade of blue, these are the genes that have the strongest evidence for this recurrent positive selection. So what we see in primates is really the vast majority of the genes that I looked at didn't have any sort of signatures of these sorts of conflicts at these interfaces. But there is this core set of genes down here that have been shaped by this positive selection, likely driven by interactions with pathogens. As we move across the mammalian groups, what we see is that this core group actually stays the same. So it's under positive selection in all 3 of the mammalian lineages that I looked at. But we also start to see a whole lot of lineage-specific genes pop out, and generally there's a lot more going on in both the rodent and bat lineages, which I think raises some interesting questions that I'd be happy to talk more about later. And so this was really pretty exciting to see because when I started this, I had no idea what we would get out. We really only focused on these sorts of conflicts at protein-protein interfaces. But what I think this data really clearly shows is that these evolutionary conflicts between hosts and pathogens extend into the realm of other types of biological macromolecules. So glycans seem to be able to also drive these sorts of evolutionary patterns. I know there's a ton of data here and a lot to process, and so there's a lot for me too, and I think this is really going to be a major source of projects in my future lab, translating this evolutionary signal into actual functional differences in the ability of these receptors to recognize and engage with microbes. But I just want to give you a glimpse into the other sorts of things that we can pull out of this data set very quickly.
Zoe, can I ask you a question?
Yeah.
Were you surprised that Dectin-1 didn't pop out?
I was a little surprised that Dectin-1 didn't pop out in primates, but it is under positive selection in both rodents and bats, which I think is really interesting. And this is something that I'm really kind of excited to pursue, to look at some variants from some of these species and compare to some of the primate species. But yeah, I mean, since it is one of the kind of major fungal receptors, I will say that when I first did this analysis, I was focused on the primates and not seeing anything there. I was kind of like, oh boy. But I think the power of these analyses and looking at multiple different groups gives us now this great resource where we have lots of things where there is diversification. And so even if it's not happening in primates, that doesn't mean that we can't learn something from it and maybe even apply it to the treatment of fungal diseases. Yeah.
If I could just follow up on that question, I mean, could it speak to the fact that maybe the receptors in primates, or there's some constraint that's not allowing variation at that interaction?
Yeah, I think that's a really interesting idea. And so one thing that I'm kind of interested in is sort of the divergence here. Is it driving specificity versus breadth of recognition? And it's possible in in rodents and bats that maybe some of these other receptors are getting specialized for fungi, freeing up dectin-1 to be able to evolve a little bit more. And so I think both looking at this on a gene by gene level, but also looking across the data set might give us the power to make some predictions like this about, why genes are constrained in one lineage but not another. Yeah, I think that's a great question. Okay, so one of the things that I did not mention is that we can use these phylogenetic tools that look for positive selection to not just pull out whole genes that are rapidly evolving, but actually to pinpoint specific amino acids within those genes that are diversifying across evolutionary time. And so I think this is really cool because it lets us start to make predictions about how these signatures of evolution are actually maybe having a functional impact on protein function. And so I wanted to ask where in these proteins the rapid evolution was actually occurring. And so to do that, I'm just going to focus on this one example down here called Langerin, which is under recurrent positive selection in all 3 mammalian lineages. And we also have a lot of structural information and functional information about how it engages with its ligands. And so what I did was I pulled the crystal structure of the C-type lectin domain of Langerin, which is what engages with its ligand, which is shown here in yellow. And I just plotted the rapidly evolving amino acids onto this structure, which you can see in teal here. And so what I hope you can appreciate is that these rapidly changing sites are really clustering around this ligand binding domain. And this is generally true for most of the receptors that I've looked at so far that we have structural information for. And so I think what this tells us is that recognition is really in the conversation here, right? So I think that these signals that we're seeing are really likely to have functional implications for either ligand binding specificity or tightness or some other kind of functional significance. So that's really cool and will be kind of a major focus of my future lab. But as we were just sort of touching on, I started all of this work trying to look for fungal-driven evolution in these host receptors. And as many of you might sort of know, a lot of these do recognize fungi, but they also recognize a number of other microbial glycans, so from other species as well. But I think there are some examples here that tell us that fungi have driven the evolution of host immune systems. One is dectin-1, which we see under positive selection in rodents and bats. But the other, and I think really interesting example, is this one down here called melanin lectin, or MELEC. This is a C-type lectin receptor, and it was recently identified to specifically recognize melanin called DHN melanin, which coats the cell walls of fungal species like Aspergillus fumigatus. And this receptor is also required for the survival of invasive fungal infection caused by these pathogens. Now, MELEC is under recurrent positive selection in both primates and rodents, and so this tells us that there's a potentially ancient role for fungi in shaping the evolution of this receptor. But it actually turns out that the story is even more interesting than that, and it seems like MELEC is evolving across multiple different evolutionary timescales. And the way I got into this was in this original paper that identified the ligand for MELEC. The authors noted that there was a fairly common SNP in human populations that occurs actually in the cytoplasmic domain of this protein that's associated with an enhanced risk of invasive aspergillosis in bone marrow transplant patients. So what they see is that individuals carrying this alanine allele at position 26 have this really increased risk of invasive aspergillosis. So I was really kind of intrigued when I went into my data set and I looked at position 26 in my primate alignments. And I saw that every primate, except for 2, humans up here and squirrel monkeys down here, carry this alanine allele. So again, this is the risk-associated allele, and glycine is protective. And so this is really kind of interesting, and I was curious if there was variability of the allele frequency of glycine across human populations. We know it's not fixed in the population. And so what I did was I looked at the allele frequency of the alanine to the glycine allele across 5 different human populations, which you're seeing on this map here. And what I hope you can appreciate is that there's really different compositions of allele frequencies across these different human populations. And so what this tells us is that changes in MELEC are happening in much more recent evolutionary history of modern humans. I think this also raises a really intriguing hypothesis about maybe the role or the exposure levels to different fungal species across these geographic groups that might be driving the skew that we're seeing in allele frequencies. But as I just mentioned, it's not just humans that have substitution at this site. There's also a substitution in squirrel monkeys. And so this is really cool because it lets us explore yet another evolutionary timescale, because we have many more extant squirrel monkey species. And so very fortunately for me, we have a whole lot of samples from different squirrel monkey species in the Aldi lab. And so what I could do is I could just go in and genotype these samples to determine what allele they have at this position 26. And so I did this for, I don't know how to get the controls to disappear. Sorry.
I think you might need to look at the chat, and then they'll just go away themselves. Okay. So the question in the chat is an interesting one, which, going back to your room model,
Yeah.
do you think that you can heterologously swap alleles across rodents to look at whether or not the evolved differences are functional?
Like in the context of an actual animal? Yeah, I mean, that would be really cool. We have not for this project, but we definitely have done things where we've engineered in kind of like different species versions of proteins of interest into mouse models. I think probably I would start with something a little bit simpler and look in cells and see if we can find differences there. So to use cell culture-based models to probe differences across these. But yeah, obviously there's lots of differences in biology, and so you always want to see it in the endogenous context. And I think rodents would be a great place to explore that.
So now if you click on that and look at it,
Okay.
the controls will go away.
Still getting used to Zoom. Okay. So hopefully they'll disappear in a minute. But so I looked at 3 different squirrel monkey species. I think they're very cute, as you can see in these pictures over here. And what was really interesting is that this valine allele, again, is not fixed in the population. It's not fixed in any of the populations that I looked at. And in fact, in these Guyanan squirrel monkeys, whose geographic range is in the Guyanas and northern Brazil, we actually see the ancestral alanine allele. And then in these Bolivian and Peruvian squirrel monkeys down here in Bolivia, Peru, and western Brazil, we see that they either have the valine homozygous or they're heterozygous for both alleles. And so I think there's a lot of really interesting questions. There's also many more squirrel monkey species that we don't have samples for currently that actually span these geographic ranges that I think would be really interesting to look at. It might tell us a little bit more about the evolutionary history here. But regardless, I think it's really quite amazing that we have this kind of convergence and substitution on this site, which I think tells us that there's likely a really functional significance to this as well. And again, this just speaks to the different evolutionary timescales over which these receptors are evolving. Okay, so going back to this data set, I think I now have this wealth of new interfaces to study, to look at these sorts of host arms race interactions. And I just want to re-emphasize that this is really a whole new territory. So we've never looked at these sorts of conflicts outside of the protein-protein context. And so now looking at these glycans and the role of glycans in shaping host immune evolution, I think we're probably going to learn some really new and exciting biology. And again, we can do this in a fungal-specific way, getting to my real interest in these host-fungal interactions by focusing on some of these fungi-specific receptors. But I'm also really excited about the possibility of asking how fungi collaborate or compete with other types of microbes to drive evolution in some of these more promiscuous receptors. Okay, so we have found these signals of evolution in the host side, and these approaches have really taken a retrospective approach. So we use phylogenetics to infer something about how evolution has happened, right? So how things have become into the species and sequences that we see today. And we can also use these types of phylogenetics or retrospective approaches for fungi, and I'll touch on this a little bit at the very end of my talk. But what's really cool about fungi and some of these fungal pathogens in particular is that they have short generation times, and we can grow really large population sizes in the lab. And so what this means is that we can also do real-time evolution. So we can use experimental evolution techniques to try to ask questions about how these pathogens are evolving and will evolve in the future. And so now I'm going to tell you a story about how I've been using experimental evolution to ask how different types of host species can shape the adaptation of fungal pathogens. And so really, I'm interested in how different host species might differentially shape evolution in fungi. And I think about this from two perspectives. So on the one hand, I'm really interested in the evolution of pathogenicity originally. So we know that many fungal species that are capable of infecting mammals are primarily environmental. So they live out in dirt and in compost heaps and on tree bark, which looks really different from inside of a mammalian host. So how did they actually gain the ability to infect these mammals? And one hypothesis that gets put forward to explain this is that these fungi are really trained through interactions with environmental hosts, so things like amoeba and nematodes, and that these interactions with these environmental predators or host species can drive the acquisition of certain traits that then lead to these populations of fungi that are then poised to be able to infect mammals. So there's something shared in life inside of an amoeba and life inside of a mammal. But then I'm also really interested about the evolution that occurs in the context of a single infection. So we know that evolution doesn't stop once these fungi get into mammalian hosts, but instead they experience repeated interactions with host immune cells, so things like macrophages. And this can also drive adaptive evolution, which has really important consequences for disease progression and outcomes. And so what I wanted to do was to ask whether we could provoke this sort of evolution in the lab and to watch it unfold in real time and use this system to ask how these different species might drive evolution in fungi. And so the system that I came up with looks something like this, where I can take environmental strains of fungal pathogens, expose them to host cells from different species, and allow them to interact over defined periods of time. At the end of these time periods, I can lyse my host cells only. The fungi are pretty tough. I collect all of these fungi. I can freeze down aliquots and create a fossil record for myself, which lets me go back and do evolutionary studies and to watch this process unfold. And then I use the remaining fungal population to repeat this process over and over again and see what I get out. Now, before I show you the data from this, I first want to introduce you to the players that I'll be talking about today. We'll start first with the host cells. So as I've already alluded to, all of the work that I've done has been in parallel in both an amoeba model as well as a mouse macrophage model. On the left, you're seeing the particular amoeba species that I work with, and I'll show you in a video here how it's interacting with these yeast cells in its environment. And so what you'll see is that these amoeba really crawl around quite a bit. They interact with the yeast, and if you're really lucky, you might capture a phagocytosis event. And so I think these guys are really fun to watch, but we also know that this particular strain of acanthamoeba was actually isolated as a contaminant from a culture of wild yeasts. So in addition to being able to make them phagocytose these species in the lab, we think they're actually doing this in the environment as well. On the right, you're seeing an image of the J774A cell line. This is a mouse macrophage-like cell line, and I'll show you a video of them interacting with those same yeast. It's very boring. But you'll see that they do phagocytose quite a lot of yeast from the environment. And so this will serve as the macrophage model for all of the work that I'll show you today. Okay, so those are our host cells. What about our fungal pathogen? If you have a really keen eye, you might already know, but the fungal pathogen that I've decided to focus on is Cryptococcus neoformans. This is an encapsulated yeast species, as you can see over here. So it secretes this really elaborated capsule structure, which prevents recognition by host immune systems. and is a member of this Basidiomycete group of fungi. So it's actually more closely related to mushrooms than it is to many of our other favorite yeasts, which are found up here. Now, Cryptococcus is really a huge problem for HIV and AIDS patients. It accounts for about 20% of AIDS-related mortality every year. And it's inhaled as a respiratory pathogen, but it causes mortality in these patients primarily through its tropism for the brain. So it disseminates to the brain and causes a really fatal cryptococcal meningitis. Now, Cryptococcus was kind of the obvious choice for me to use for this system for a number of reasons, but one of them was the fact that we have really a ton of community interest in sampling cryptococcal isolates from diverse locations. So this phylogeny here is from John Perfect's lab at Duke, and contained within it are about 400 different strains that came from patients and clinics, as well as from environmental sources. And so what we see from this is that there are really kind of 4 major lineages of Cryptococcus neoformans, and there are various levels of genetic variation as indicated by these branch lengths and different environmental reservoirs across these lineages. And so I think this really provides an incredible resource for starting to ask questions about how phenotypic differences might map onto genotypic differences, and I'll come back to this, and that can tell us something about the evolution of this species. But I also saw this as an incredible resource for finding a strain to start this experimental evolution. And so what I did was I got about 35 of these strains from John Perfect. They encompass all of the major lineages and are primarily environmental isolates. And I got these into the lab, and the first thing that I wanted to do was to ask how well they're able to replicate in our 2 host cell models. And so before I show you that data, I just really quickly want to walk you through how I do this experiments, because this will be most of the data that you'll see today. So what I do is I take a Cryptococcus strain of interest, I put it into culture either with macrophages or amoeba, and I allow the crypto to be phagocytosed over the course of 1 hour. After that 1 hour is up, I remove everything that remains extracellular, so anything that hasn't been phagocytosed gets thrown away. And then I replace the media and allow everything to grow together for an additional 24 hours, at which point I lyse the host cells, collect all of the fungi, and plate them so that I can count CFUs. And importantly, I also plate a series of controls after this initial 1 hour phagocytosis step, which gives me a starting input count. So now what do these strains look like when we use these co-culture experiments? So we'll start first with a subset of these strains and how well they're able to grow in macrophages. On the y-axis here, I've plotted the log2 of the CFU fold change. So this is that 24-hour count divided by the 1 hour count, where a value of 1 indicates a rough doubling in the host cell. And so what I hope you can appreciate is that there is strain variation across these 8 strains that I'm showing you here, where some seem to be better and some seem to be worse. But on the whole, most of these strains are actually surprisingly good at replicating in macrophages. The picture looks really different if we look instead at amoeba. So this data is plotted in the same way, where the y-axis is that log2 CFU fold change. It's the same set of strains here. And so what you can see is that most of these are falling below the axis, indicating that the amoeba are really killing these strains quite well. There is, again, some strain variation across these, with some of these strains actually seeming to be somewhat resistant and capable of replicating. Yeah.
Are the macrophages activated in any way?
Yeah, so I do use activation. I use PMA to activate, but I've also looked at activating with interferon, and I see roughly the same, yeah. And I guess for the aficionados, all of the Cryptococcus are also opsonized in order to facilitate this. Yeah, so there are some really interesting strain differences that are coming out, and this is something that I'm interested in continuing to push on in the future. But in the interest of time today, I'm just going to focus on this one strain here called FTC555-1. This is an environmental isolate that came from a mopane tree in Botswana. And as you can see, it has a sort of decreased ability to replicate both in our amoeba model as well as our macrophages. And so it served as the starting strain for my experimental evolution. And so what I did was I took FTC555-1, I threw it into this serial passaging scheme, which looks really similar to what you've seen on previous slides. There's one key difference, which is that I couldn't actually recover enough fungi after a round of passaging to repeat it immediately, and so there was an outgrowth step included here. And I did this passaging for a total of 12 rounds. I independently evolved 6 populations. 3 of these were passaged through amoeba cells, and 3 were passaged through macrophages. Now, in the interest of time, I'm not actually going to show you any of the amoeba passage data today, but I will say that I think it really beautifully illustrates the power of this system to reveal different evolutionary trajectories across different populations. So we actually see really different things coming out from our 3 independent strain lineages. But instead, I'm going to focus on the macrophage data. And the first thing that I wanted to ask was, I've passaged this fungus through macrophages for 12 rounds now. Do they actually grow better in macrophages? And so that's what you're seeing here. So this is the same sort of experiment I showed you before. On the left, you're seeing our parental strain. So this is what we started with. And these 3 shades of blue, indicated by these M strain names, we can see that in our 3 macrophage passage populations. And so we can see that in 2 of the 3 independent lines, we have this really robust enhancement now of these strains to replicate in macrophage cells. I was really interested in whether this might also confer an advantage in another host species. So I took these same strains and I put them into our amoeba model. And when I do this, what I see is that there's really no change in the ability of these strains to replicate in amoeba. So again, the parental starting phenotype is here on the left. And if anything, this M1 strain might have gotten a little bit worse at replicating in amoeba. And I think this raises some interesting questions about trade-offs that might be associated with adapting to one specific host type.
Zoe, did you look at other host cells, neutrophils or DCs? Or?
I haven't looked at other types of host cells. I have looked at other macrophage cell lines, and I do generally see the same thing for these in other macrophages as well. But I have not looked at human macrophages yet. That's one thing on my to-do list.
Maybe you're going to get to this, but do you think that the result is ROS dependent or ROS independent?
Ooh, I don't know. That's a good question. I haven't really, so as we'll get to it, I haven't really looked at the host responses here yet. I think that's a really interesting question, and it's something that I'm trying to do right now. But I've mostly been focused on the fungal side for now. But I think we now have these strains that have very different phenotypes that we can use to probe changes in host responses, which is something I'm really excited about. Yes?
So you're not selecting for fungi that avoid phagocytosis, right?
No. So yeah, that's a great question. So I have looked at phagocytosis rates across these strains. I don't see any change in the phagocytosis of the evolved strains versus the parental. They all seem to get phagocytosed to the same level, both in macrophages and in amoeba. Yeah, so I don't think it's a change. I don't think it's a huge change in cell wall structure or something that's affecting opsonization or any of that. Yeah. Yes?
This is really cool. Have you seen any differences in like growth rate of your full populations or?
Growth, yeah. So a little bit. So if I look in a rich media like YPD, I don't see a huge change when I grow these in isolation. I have done a lot of competition experiments. I'm not going to show any of that data today. And I see that this M1 strain, which is really what I'm going to focus on for the rest of the talk, does outcompete the parental in the macrophages. But interestingly, in YPD, the parental strain actually outcompetes the M1 strain. And so again, I think there might be some trade-offs here in terms of where you're happiest growing. But if I just look at them growing by themselves, I don't see any difference, which I think is kind of interesting. Yeah.
To follow up on that, it's interesting how the M2 doesn't have that phenotype. Does that carry on with the other assays you're talking about?
Yeah, so I haven't used M2 in a ton of my assays, but it is this really nice control where it was passaged through these cells for the same amount of time and doesn't show the phenotype. And so, yeah, I think it's really fascinating, and I think, again, kind of speaks to the power of this system where you're going to get different things out in every population that you subject to this sort of passing approach. There was another question. Yeah?
Hi. No, this is super cool. I'm curious, like looking at the amoeba videos, it looks like it wasn't a particularly large fraction of the fungal population that's phagocytosed.
Yeah.
And you mentioned that you got a lot of different outcomes, which is suggestive to me of drift rather than selection, that you're basically bottlenecking the population and getting a lot of different outcomes that maybe aren't actually related to selection by amoeba. Is that a possibility, or am I just being too worried? Or
No, I think that's a fair possibility here. The other thing that I'll say is that, as I showed with the data, the amoeba exert a much stronger selective pressure on the population. So in addition, those are all corrected for how much gets phagocytosed, right? So in addition to there being lower phagocytosis rates, you're also seeing less replication. And so the number of generations in these experiments for the amoeba is way, way lower than for these macrophages. And so I actually think I'm amazed that we got anything out of the amoeba passaging experiments, and I'm excited to see what's going on there. But I think for those sorts of experiments, carrying them on for much longer might give us more non-drift and significant events. Yeah.
So a question in the chat, which you might want to hear, is asking about whether or not you see the same trade-off between amoeba and macrophages, where if you evolve them in the amoeba, do they have a less fit in macrophage?
Right, yeah, so I have looked at that. I'm not showing it. I can pull it up later if people are interested, but I see exactly the same thing. So I do see that there are amoeba passage strains that are doing better in amoeba, but when I look at them in macrophages, actually there is absolutely zero change in their ability to replicate in the macrophage system. And so I think in this particular experiment, what I've done is I've really host adapted these strains to the host species that they were passaged through. And I haven't selected for any sort of generalizable pathogenicity related trait. But yeah, it's a great question. Yes?
Outside of replication, did you identify changes in other pathogenicity traits like melanization?
Yes, I will get to that. So hold that thought. Okay, other questions? I'll move on then. Okay, so for the rest of my talk, I'm just going to focus on my efforts in trying to understand one of these strains in particular, so this M1 strain. I've done a deeper dive on trying to get down to the mechanism of how this strain actually gained this enhanced ability to replicate in macrophages. And so what I did was I took a number of single colonies from the end of my passaging. I also took the full pooled culture and I sent them out for sequencing. And when I got the data back, I was really kind of surprised to find that in these different samples, there was only a single mutation and it was fixed in all of these different samples. And that mutation is a single nucleotide change that causes a missense mutation in a gene called CAC1. Now, CAC1 in Cryptococcus encodes the adenylate cyclase gene. So this is required for the production of cyclic AMP and kicks off this cyclic AMP signaling cascade, which is required for the production of many virulence factors, such as capsule and melanin. It also controls things like mating and cell size, and many other really important aspects of cryptococcal biology. And so the fact that this was really my only candidate mutation, combined with the central role of this pathway in regulating virulence in this species, really suggested that this was probably my causative mutation. But I wanted to confirm this experimentally. And so what I did was I took my parental strain and I introduced this single nucleotide change in the endogenous locus. And when I do this, I see that this is sufficient to drive this enhanced replication in macrophages up to the level of my M1 evolved strain. I can do the reciprocal swap and take the M1 strain and put back in the parental allele of CAC1. When I do this, this abolishes the ability of these strains to grow as well in macrophages back down to the level of parental. And so I think this really nicely illustrates that this particular mutation is both necessary and sufficient to drive the phenotype that I'm seeing in my evolved strain here. The next thing that I wanted to do was I told you that I can freeze down this aliquot to create a fossil record. And so I wanted to use this fossil record to try to understand the evolutionary dynamics of this allele over the course of my experiment. And so what I did was I went back and I pulled out all of my frozen aliquots. I started cultures from these and sent them out for sequencing. And then I just asked what the allele frequency was of this CAC1 allele over time. And so on the y-axis here, you're seeing the allele frequency of the evolved CAC1 alleles over the course of my 12 passages. And so what you can see is that this mutation arose de novo in the 4th passage, and then very, very quickly swept through the population. So by passage 6, it's basically at 100% in the population. And so what this tells us is that this mutation really confers a strong fitness advantage for growth in macrophages. And in data that I'm not showing you, looking at the phenotypes of some of these intermediate passages in my experimental model, I see that this really nicely correlates. Okay, so we know what the causative mutation is. We can watch how it swept through the population over time. So what is the actual mechanism here? And I'll tell you, I'll spoil the punchline a little bit and say that I don't actually know. But I have started to look at some of these downstream virulence factors, which I'd be happy to talk more about later. But right now, I'm just going to tell you a little bit about what we can learn from using some more evolutionary approaches as well as structural modeling about the nature of this allele. And so one thing that we can do is we can look at conservation of this particular amino acid across both fungal species as well as across cryptococcal isolates. And so first, looking at fungal species, if we look at the adenylyl cyclase gene from a number of different fungi, including both pathogenic and non-pathogenic species, what we see is that largely the adenylyl cyclase is not highly conserved. The average pairwise identity is about 30%, but again, this is really hundreds of years, millions of years of evolutionary time, so it's not super surprising. But if we do look at this arginine, which is the site of the mutation, what we see is that this arginine is actually conserved in a lot of these different species. And so I think this starts to hint that this might be really kind of a functionally important site for the function of this protein. Interestingly, if I look at the more than 500 different isolates of Cryptococcus neoformans that we have sequencing data for, I see absolutely no variation at this position. There is no mutation happening there, despite the fact that there's quite a bit of variation across the rest of the protein. So again, I think this starts to point at possible trade-offs associated here. And really, to me, this suggested that this was likely to be kind of an obviously important residue for the function of this protein. And so to try to get at this, I used structural modeling. I used AlphaFold to look at where this residue falls in the CAC1 structure. And so that's what you're seeing here on the right. I've indicated some of the major functional domains of CAC1 in these different colors. So it's regulated partially by RAS, and it has this RAS interacting domain down here. The catalytic domain that produces cyclic AMP is shown in purple. And then all fungal adenylyl cyclases contain this leucine-rich repeat region, which is characterized by this kind of saddle-like structure. And this is thought to sort of coordinate interactions with lots of other regulatory proteins, but we don't know a whole lot about it. And so I really thought we were going to see that this position was somewhere in the catalytic domain, affects the production of cyclic AMP. But instead, when we look at where it falls, it sits right here, kind of sticking out of one of these leucine-rich repeats. And so this is really kind of crazy to me. I would challenge any one of you to look at this structure and pull out that particular residue as being functionally important. But I think what this really tells me is that the system has the power to reveal interesting variants that nature has not revealed to us that we can then use to learn something interesting about the biology of this species and its ability to replicate in different types of hosts. And so that's all I'm going to tell you. I'll be happy to talk to you about melanization and capsule at the end if you want. But I wanted to close with kind of one last experiment that I'm sure many of you are wondering about, which is that I've done all of these experiments in vitro in cell culture. So what does this strain actually look like in the context of an actual infection? And so what I did with the help of two graduate students in Jessica Brown's lab at Utah is I took both our parental strain and our evolved strain, and I infected mice with these strains and simply asked how those animals were able to survive infection. And so that's what you're seeing here on this slide, and I don't think you need me to tell you that there's a big difference here. So until you're seeing our parental starting strain, and when animals are infected with this strain, they do develop symptomatic disease and they eventually succumb to it. In contrast, if we infect animals with our evolved strains, this is the strain that does so well in macrophages, we see absolutely no signs of disease, and as far as we've carried out these experiments, we never see the animals succumb to infection by this strain. And so I'll be honest, I initially saw this and I was very disappointed because generally, macrophage growth has really well correlated with in vivo outcomes. And that's kind of the opposite of what we're seeing here. But one thing that I was really curious is whether this M1 strain is simply being cleared, or if perhaps it's kind of persisting and sticking around in an interesting way. And so what we did was we took all of the animals at the endpoint of this experiment and we collected both their lungs and their brains, and we plated them to see if we could recover any Cryptococcus. And so that's what you're seeing in this plot here on the right. And I was really surprised and kind of delighted to find that we do actually recover high levels of Cryptococcus in the lungs of all of these animals infected with the M1 strain. And we also see dissemination to the brain. And so I think this is really interesting and gets back to what's going on the host side here. And so what we know is that this strain is capable of establishing a persistent infection, but is it somehow modulating host responses to be better tolerated in these animals? And I think this is a really exciting kind of future direction of this project that we're sort of actively working on right now.
Zoe, so one of the big mechanisms, for crypto to disseminate is to get out of the macrophage through this process called vomocytosis. So did you select for a strain that can't do that, and now they just replicate within the macrophage?
Yeah.
Have you looked at that?
So I haven't looked at that directly. It's a little bit hard. So one of the things that I wanted to do was to just prevent vomocytosis and see if that kind of changed the outcomes entirely. It's much easier to trigger vomocytosis than it is to prevent it. But what I can tell you is that if I look at my populations at the end of my macrophage experiments and I separate extracellular from intracellular, I see roughly the same percentages of extracellular cells in those that have been exposed to M1 as opposed to the parental strain. So I don't think there's a huge vomocytosis, but I think that's a really interesting question. Yes. So we actually just got the histology back, and I don't really know what I'm looking at. So we're working to get someone else to score it. But we see tons of crypto, and we did the histology at an earlier time point, so before the parental animals started to go down. We see tons of crypto in the lungs of both animals, and there's nothing totally obvious to us in terms of the different distributions within cells or anything like that. But it's a great question, yeah.
I just can't remember if the animal model recapitulates the human disease in that it's the infections in the brain that leads to death in most animals, or
Yes.
it's the lung.
Yeah, so usually death is correlated with high brain dissemination. So I think the fact that we're seeing dissemination to the brain is really interesting, and we see it in the majority of the animals. I think there was one in this experiment that we didn't recover any. But yeah, so normally that's correlated. I will say that when these animals are succumbing, so we did also look at burdens in some of these animals that were going down. It's way higher than what we're seeing here. So we normally see at endpoint somewhere around what we see in the lungs, in the brain. Yeah. Okay.
Did you look at granuloma formation? Is that
Yeah.
between the two, or?
Yeah, so that's a great question. So in human patients, we see granulomas form after Cryptococcus infection. That is generally not a feature of mouse infection, but this is something that I think the field has been really interested in, and there have been a couple strains that have been identified where you do see granulomas. I am pretty sure from looking at the histology that we're not seeing granulomas. So I don't think that's going on here, but again, I think we need to have someone who knows more than I do about mouse lungs to look at that and score it for us. Yeah.
Any chance you re-challenged those mice, the M1 mice, with the wild-type crypto strain?
I did not do that. That's an interesting idea. Yeah. Yeah. And see if they're protected. Is that the idea?
There's a lot of interest in vaccine development, right? So some of Amaryllis Rivera's work with crypto and Stu Levitt's, and it's a pretty remarkable result. So.
Yeah. We also have some cytokine data, which I'm still working through. If people want to talk more about that offline, I'd be happy to do that as well. Yeah.
I guess if you immunosuppress them, do they then reactivate?
Right. So this is we actually just did this experiment. And so we only did it in M1 infected animals, and we immune suppressed them. They started to decline, but they didn't. When Jessica's group has done this before, they've seen really rapid declines within like a couple days of immune suppression. These animals did not. And so we did end up taking them down and looking at burdens in those, and we do see more in the immune suppressed animals than in non-immune suppressed animals, but it was kind of surprising to us. Yeah. All right. Okay, so that's my Last slide on this. So just to summarize the fungal side, I've developed this serial passaging system, and we can use it to select for Cryptococcus populations with enhanced survival on different types of phagocytes, which I think is really cool. And then I told you a little bit about how we can go down to mechanism of how this happens by exploring one particular macrophage-adapted strain where I recovered this mutation in this CAC1 gene, which confers this enhanced growth in macrophages. I can watch the evolution unfold, which I think is really the coolest part about this. And then really interestingly, we see that this strain just establishes persistent infection, but kind of in the absence of any signs of disease. And then as we've kind of already touched on, there's, I think, a whole lot of interesting future directions from this work. I still don't know what the mechanism is, and that's kind of driving me crazy. So that's one of the big focuses of my work right now. And then, of course, what's going on the host side? I think that's the big thing that's missing from this story. How do host responses differ between the evolved and parental strains, both on the cellular level, but also in the context of animal infections? And then last, I have all of these other populations, so both from the amoeba, but also from the macrophages, and what does adaptive evolution look like in those populations, and how is it different from what we see here? And so hopefully I've convinced you that taking these evolutionary approaches can reveal some really exciting biology at these host fungal interfaces, and I think have really brought us into a new realm of evolutionary conflict that I'm excited to keep exploring in my future lab. And so I'll continue to push on both sides of these interactions, and just to give you a brief glimpse of what I'm planning to do, I think the big focus on the host side, as I've already said, is really in trying to translate this evolutionary signatures into these functional differences in the ability of these different receptor variants to engage with microbial species. And so again, I'm excited to do this in a fungal specific way, but I'm also really excited to explore these questions of kind of polymicrobial interactions and how they might have collaborated or competed to shape the evolution of these receptors. And I think this has been something that we really haven't been able to explore before by studying conflicts at these protein-protein interfaces that now this glycan world has opened up to us.
Zoe, have you genotyped the J7-4 receptors here? Do you know the different alleles?
Oh, no,
and then compared that to different mouse strains?
No, I haven't done that. That's a great idea.
Yeah. I'm just thinking if you could take your M1 population and look at a different mouse strain that has a different allele for, say, dectin-1,
right?
Do you then reverse the phenotype?
Yeah, no, I think that's a great idea. I mean, there definitely are strain differences across mouse strains in cryptococcal susceptibility, and I will say there have actually been a couple groups that have looked at FTC 555-1 in a different mouse background, and they see a very different virulence profile there. And so some of this is we're using C57 Black 6, which seem to be a little bit more susceptible. But yeah, comparing the alleles and trying to bridge projects would be a really cool.
I wonder if there's a huge, divergence between the J77s and the C57 background. That could possibly explain the result that you got.
Yeah, that's a great idea. Yeah.
So do you have any guess or hypothesis why CLEC9A, for example, is so under pressure in the rodents and in the bats? The MRC receptors are under so much pressure?
Yeah, I think it's a great question. It's hard to know. I do think, so CLEC9A is actually kind of interesting. So some of these I threw in, they don't actually seem to bind necessarily to microbial ligands, but they have self ligands. And so initially I threw them in as controls, essentially. So things from the same family that we don't think should be shaped by these host pathogen interactions. And so why are they coming out here? And some of this, I think, again, could be differences in immune system function across different species. So it could be that in primates, we don't really use this, or maybe even in mice, we don't use it to detect pathogens, but in other rodent species, you do. And so the signature could be driven by certain species within those lineages, and that's what's reflected here. But yeah, I mean, I think some of these, I think initially I'm kind of more drawn to the ones that have more signal across these, because then we can do lineage comparisons. But it's a really interesting idea about these ones where we see it only in one specific lineages. I think bats in particular are really exciting to me because we know that bats are kind of experiencing their own pandemic with white nose fungus currently. And so I think also their kind of geographic location in caves, they're probably exposed to many more fungi, but also we know that they're exposed to tons of microbes generally, and their immune systems are kind of crazy. And so I think this signal is really exciting, and I think there's a lot to explore there. Yeah?
I was curious what is known, if anything, about the level of diversity of some of the glycans that are recognized by these. Is it something that changes within a species, or is it more broadly diverging across species for a given? I imagine that hasn't been profiled to the same extent because it must be much harder to.
Yeah, so this is actually something that I'm really interested in, and a lot of the studies of these have been done kind of in the context of ligands taken out of the microbial cell wall, so not in the context of an actual microbe. And so I think in addition to using these methods like profiling ligands, we can learn something, but I think the more powerful method will be to actually take the whole microbe and see if there are differences, because I think the context changes a lot when it's in a cell wall versus when you take it out. And then I think building in some of these collections of strains from both clinical isolates, environmental isolates within a species, I think there is likely to be quite a lot of variation. I know in fungi we do see that. I think probably in bacteria that's also happening. And so part of my plan is to not just try to identify the single species that's recognized, but also to try to identify the ligands, and to do this using a combination of genetics, but also profiling large libraries of microbes. Yeah. Okay. So these are my plans on the host side. On the fungal side, as I mentioned, I'm also interested in using these phylogenetic and genetic approaches to try to understand how these strains in this phylogeny have evolved into the strains that we're encountering today. And so as I showed you earlier, there is phenotypic variation across these strains. This is kind of an expanded panel of how some of these strains behave in our amoeba system. And I think there are some really exciting leads to follow on here. So some of these strains seem to actually be really good at replicating in amoeba. And this sometimes these also correlate with enhanced replication in macrophages, which might start to let us get at these questions of kind of shared traits of pathogenicity across species. But then I've also developed tools to speed up our ability to phenotype these strains in the hopes that we can get down to kind of the genetics of what's driving these phenotypic differences. And so that'll be a major focus of my lab as well. And then last but certainly not least, I'm super excited about these serial passaging approaches. And now that I have the system up and running, I think there are many ways that we can perturb it and ask how that changes this evolution. So we can imagine that changing the starting Cryptococcus strain might have a really profound effect on the evolutionary trajectory that we see. But we can also imagine that changing these host cells could also influence what we get out. And so looking at macrophages from different species or with different alleles or from different tissues, I think would be really interesting. And then I'm also really excited about the role of environmental host species in shaping fungal evolution. And so using amoeba and other amoeba models, as well as maybe getting back to my C. elegans roots and building in some C. elegans host fungal interaction work. And then last, I think one of the kind of major things that we think about that prevents fungal infections from really taking over and causing a whole lot more mortality in humans is their ability to evolve thermotolerance, so to tolerate human body temperature. But I think as our world is warming, this raises the really interesting question about the role of environmental changes in driving the emergence of new pathogens. And so I think we can use similar serial passaging systems to ask questions about how thermotolerance might be correlated with the emergence or evolution of pathogenicity in this species. And so with that, I will just thank all of the people that made this work possible. My mentor, Nels, has been a great mentor over the years. Mara Schwiso is a technician in the LD lab who has been working with me and had her hands in a lot of the data that you saw today. John Perfect's lab sent us all of those Cryptococcus strains that you saw. And then Jessica Brown's lab, and in particular Joe and Crystal in her lab, have been helping me with all of the mouse experiments that you saw. And then funding, so my funding comes from the Helen Hay Whitney Foundation. And again, I'm part of this Leading Edge community, which is a really amazing community for women and non-binary postdocs that I'd be more than happy to talk to any of you about. And I would be happy to take any more questions. Yeah?
That was a great talk. I'm wondering, have you looked back in the initial strains that you tested in Cryptococcus, which is the ones that were really good at replicating in macrophages, to see if they had that same CAC-A allele SNP?
Yeah.
And they did.
Yeah, so I don't see that particular CAC1 mutation in any strain. And I don't actually see any mutation at that site in any strain. But one of the things that I think is really interesting is that there's quite a bit of variation across CAC1 generally. And so for the particular strain that I'm working with, it's actually, I think there's about 20 amino acid changes, including in functionally significant domains from the reference strains that we study in the lab. And so something that I think studying these kind of diverse strains allows us to do is to ask questions about how these kind of basic biological pathways are functioning differently across these different strains and how that kind of plays out in terms of pathogenicity.
Do you think you could make one of the best strains even better at being at replicating itself in macrophages?
Yeah, I think that's an interesting question. Something I also think a lot about is maybe this strain was particularly poised to pick up a mutation there or in that way. So is there something about the allele of CAC1 in this strain? And so introducing this allele into other strain backgrounds, other alleles across these strains, I think could be really cool and could tell us something interesting about cyclic AMP signaling and virulence. Yeah. Yeah.
So, really cool work. I'm wondering if I'm making the correct assumption that based on the fact that this bug is a big problem in HIV infected individuals, that there's an important component of T-cell dependent immunity.
Yes.
To this, so when you were talking about, when the question came up, if you've immunosuppressed the animals and looked for reactivation, what is that mechanism of immune suppression?
Immune suppression? I think they were using steroids. I shouldn't, yeah. We could also look at it, and I know that they have a T-cell knockout mouse, and so I think looking at it in that context would be really interesting.
Because I guess I was wondering if potentially what you could be looking at could also have to do with regulation of antigen presentation, right? And then building on that, I was wondering if you thought about maybe adapting the in vitro evolution system instead of just looking at growth of the fungi to some sort of functional innate immune readout.
Yeah.
To basically sort on macrophages that are, for example, based on either reactive oxygen species levels or production of a type 1 interferon response or whatever you think is important, right?
Yeah, no, this is like my dream. So to be able to like both do the evolution, but also to watch host responses in real time and maybe even to select on that, I think that would be really cool. I haven't figured out how to do it yet. So if you have ideas, I'd love to talk.
Sure, and then one last question would be, have you thought about also trying to do the evolution in vivo in the animal model? Like as the parental strain infected animals, like before they start dying, right?
Yeah. Yeah, so people have actually done some serial infection. And I think there have been sort of mixed results with that. I think I'm also kind of more interested in these like repeated interactions with immune cells and not necessarily like sacking the whole animal and collecting all of the crypto out. But I think it's an interesting idea. And I guess one thing that I haven't mentioned is that when people have done this serial passaging through animal models, we do actually, there have been some mutations recovered in the cyclic AMP signaling pathway. So I think we're also seeing some kind of convergence onto this pathway as maybe a hub or a hot spot for evolution. But yeah, it's an interesting idea. I think my expertise is not so much in the mouse side, and so that's why I've stayed away from it for now, but I'm not opposed to trying it in the future. Yeah.
Great. I think one more question.
Okay.
But with your cellular passaging, you had those 3 populations that arose in the macrophage. And I think it was M3 that was the next highest.
Yeah.
Have you gone forward with sequencing that and trying to figure out what pathway? Is that the CAN pathway as well, or is it an entirely different one?
Yeah, it's an entirely different one. I don't know. So the evolution in that population, spoiler alert for my chalk talk, it looks really different. So we actually see it's a much more heterogeneous population. It seems like there are multiple different kind of flavors of Cryptococcus in there that are carrying multiple mutations. And I think that's going to be a really fun system to go back and to look more at this kind of evolutionary project, how it proceeds over time and how it acquires mutations, whether it was sequential or kind of they're not linked, so I don't think they arose at the same time. But yeah, that's like something I'm really interested in. And there, it's not CAC1. So in all of the other strains, I can tell you I've genotyped all of them. I see no mutation in CAC1, so it's unique to this population. Yeah.
Great. Well, thank you very much.
Thank you.